or was a senior scientist with the Kansas Geological Survey. He received his BA in geography from the University of Texas at Austin, an MA in geography from Kansas University, or University of Kansas, and a PhD in special studies with a focus in quaternary geology from University of Kansas. He spent most of his career studying earth sciences, processes, especially the evolution of river valleys and soil genesis, and is the former editor of Geoarchaeology and International Journal. So I'll just start at the top.
That's a good place. Thank you. So as was mentioned, my training is out of the geosciences. Even though I'm a professor in the Department of Anthropology, I'm actually still a senior scientist at the Geological Survey. And I was director of the survey for about 6 years until August, and I moved back to my non-administrative position. I'm a much happier man because I'm doing more of what I'm going to show you tonight. I've really had the fortune to work at some really interesting sites throughout the world, some world-class sites like Ain Ghazal in Jordan, Watson Brake, oldest earthworks in the Americas, worked there, and throughout the mid-continent, some key sites. And more recently, as you see here, the Bluefish Caves. I'm going to talk a little bit about the Odyssey program. And if nothing else, you'll understand how difficult it is to find the early record, because I'm going to focus on that. Okay? So bear with me before we get into talking about Bluefish Caves. So I'm going to take you back to the Folsom site, 1927. So before Folsom was discovered, there was actually a lot of pushback from the scientific community, including the Smithsonian Institute. Now, there were some premier scientists there like Hrdlicka, who said there's no way people lived with extinct Pleistocene fauna, right? And so Folsom discovered in 1927, and guess what? You can actually see this. They literally cut it out of the site and took it to the Denver Museum of Nature and Science. And there's a Folsom projectile point right there with the remains of extinct bison. So that changed that paradigm, right? Paradigms are hard to break, but it broke that one. People weren't living at the time. Pleistocene animals were roaming across the landscape. There's what a Folsom point looks like. I suspect many of you are familiar with it. And look at the radiocarbon ages, both in calibrated 12.6 to 12.1 or 10.7 to 10.2, approximately in uncalibrated years before present. And then, of course, not long after discovery of Folsom was discovery of Clovis. And there's the gravel pit where it was discovered, the South Bank, and lo and behold, here's a mammoth vertebra right there with a Clovis projectile point almost in direct contact with the vertebrae. And so this then pushed the presence of people in North America even further back in time. So 13.5 to 12.8 in calibrated years, or 10.6 to 11.6 to about 10.7 in uncalibrated years BP. So, we're starting to realize the hunt was on. This really launched the hunt for Clovis and Folsom in the Americas. All right, so let's jump from the early 1900s, and talk about Pre-Clovis, okay? So Nature, the journal Nature, March 1988, Monteverde, 14.5 to 1400 in calibrated years BP. So this is pushing it back over 1,000 years before Clovis. And when you start looking at where Monteverde is located down in the southern tip of Chile, you have to start thinking that if people were down here at that time, it's a pretty long walk from coming across the Bering Strait, down through North America, down through Central America, to practically the tip of South America. So you got to think people had to have been further north before 12.5 in radiocarbon years, closer to 14 in calibrated years. Okay, let's keep going. So Science, Journal of Science, 2013, Paisley Caves, Western Oregon, 14.5. So we're going further back in time now. Okay, this started raising, there was pushback from a number of Clovis first people saying there's got to be problems with this site. And initially there were, but those have been mostly resolved. Let's keep going. Science, March 2011. The Friedkin site, Central Texas. Also the Galt site, which is right next door to it. I heard Mike Waters was here giving a talk, and maybe you heard a little bit about these sites. 13.2 to 15.5 in calibrated years BP from the Friedkin site. 16 to 20,000 years ago for the Galt site. Of course, this has also stirred up a lot of controversy. Well, let's keep going. Science, August 2019, Cooper's Ferry, 16,540 to 15,280. This is in Idaho. And not more recently, Science, September 2021, the White Sands National Park, New Mexico. How many of you have heard about this site? I'm just kind of curious or familiar with it. Okay. 23 to 21,000. And here are the artifacts. The artifacts are actually human footprints. I think you can see pretty well, some people initially said, oh, those were ground sloths. I don't think so. I would say the archaeological evidence is irrefutable. These are human footprints. Now, I will tell you that there's been pushback on this site too because of the radiocarbon dating. What they did was these were in gypsum dunes, which is why it's white sands. They're very white dunes that cap lacustrine type deposits, shoreline deposits from an old lake that's not there anymore. And they got the seeds of a kind of a hydrophyte type plant above these footprints and below it. So they have bounding ages. The radiocarbon ages are not questionable in terms of their context. They're in place. The problem that's been articulated is that they are hydrophytes, and some people claim, well, you're going to have what's called a reservoir effect in the seeds. In other words, old carbon that's absorbed by these plants ends up in the seeds, and you can't really remove that carbon. The problem, the counter to that is Vance Holliday, who's the geomorphologist on this project, has pointed out that the geomorphic setting is really a toe of an alluvial fan, and these plants were actually getting their moisture from meteoric water, in other words, rainfall that's kind of flowing down this alluvial fan. Meteoric water does not have a reservoir effect. So it's not going to be affected like you do when you're dealing with plants that are actually physically living in a lake. So the reservoir effect can be a problem, but in this geomorphic setting, probably not. But this battle is raging on because, now we're talking the last glacial maximum, the LGM, with those dates, okay? Okay, so now we're going to get really serious. Nature, April 2017. Nature's a pretty good journal, right? The Cerruti Mastodon, Southern California, 130,000 years ago. How many of you have heard about this site? I'm just curious. A couple people have. Okay, so the first thing I got to tell you is that Stephen Olin, I was on his PhD committee, and I've worked a lot with Stephen Olin. Jared Beaton, who's listed as a co-author, was actually my very first PhD student. And so I always told them to think outside the box. This is thinking way outside the box. The site was actually, you can see it here in this image. It's literally in San Diego. It was discovered along the shoulder of a highway when they were building the highway. And it was excavated as a paleontological site, not as an archaeological site. So Steve Holland was looking at the assemblage. It was in the museum. Steve has done a lot and written a lot about bone modification. How he looked at particularly at sites in Eastern Hemisphere where you get a lot of sites where there are no lithic artifacts. You just have bone artifacts. So he's very versed in understanding what bone artifacts look like And he has claimed that these are artifacts, these bones that were found in association. Here's one of the bones of a larger bone of one of from the mastodon. And you see these rocks, these stones that are sort of scattered around it, that those are hammer stones and anvil stones used to break up these bones, okay? 130,000 years ago. So we don't even know whether those are Neanderthals, hominid, what are they? That's problem number one. It was dated with uranium series dating. In all likelihood, it's older than 130 because bones are open systems, and I got to think that typically when you have that situation, you typically get older dates from uranium series than that. That's not the real age, but it's in the ballpark. So there's a lot of, needless to say, the geneticists hate this site. Okay, they really, because of the genetic clock, if you look at the indigenous people of North America, then this 130,000 doesn't work. However, if it was a founding population that didn't make it, didn't survive, maybe it is real. So it was interesting. So I had a call when the article came out in Nature that day. I got a call from the journalist from the New York Times. It said, "So what do you think about the site?" I said, "Well, I told him what I just told you." I said, "Two of my students were on this, so I'm not sure you want to hear what I had to say, because he said, "Oh, no, that's fine. You can tell me." So I had to choose my words carefully because I knew I was going to piss somebody off, regardless of what I said. So I was very careful, and I said, I never use the word impossible. I tell my students not to use that word. I do not use that word. It's possible. And the reason I say that is because continental drift was considered impossible by some pretty distinguished geologists into the 1960s. And it was proven, eventually that it's real, right? So all those scientists who said impossible, had to eat their words, right? So which is why I really avoid that word. So I kind of left it at that. And of course, in the New York Times the next day, it says, University Distinguished Professor Rolfe Mandel says it's quite likely. So there's a lesson there, okay? I'm still learning it. So anyway, but it's intriguing. It's not impossible. And actually, the Australians have really grasped onto this because, they used to be that they didn't think Australia was inhabited until the very end of the Pleistocene, early Holocene, and now we know that people were there 50,000 years ago. So they're not bothered by this at all. But there clearly are some issues with it. I will tell you that. That's all I'm going to say about it. Okay, so as was mentioned, I'm the director of the Odyssey Archaeological Research Program. This was established by Joseph and Ruth Kramer. It's an endowed fund. It's housed at the Kansas Geological Survey. And it's actually now the largest endowed research fund at the University of Kansas, which is I think that's remarkable that something like this would be the largest. So, it provides resources to do things that normally something like NSF, National Science Foundation, would never fund. What I refer to as high-risk projects, where you're not really sure whether you're going to get a positive result from what you're doing. NSF always wants you to test it first, then come back and say, yeah, it is. And if it is, they might fund it. I have done that. And they said, well, you've already figured it out, so we're not going to give you any money. So, but Joe Kramer, he had this in mind. He said, I don't want you wasting years trying to go after funding because it takes a lot of time to write a proposal. And so basically said, here's this money, go out and do it, just do it. And so every year I develop a plan and then I send a team out, I go out with them. So, but Joe Kramer was a geologist, and so he really pushed, and for me, this was not a difficult push, to use geoscientific methods to search for the material remains of the first people to occupy North America. There's a strong emphasis on finding pre-Clovis cultural deposits, and ideally older deposits, cultural deposits. And so to kind of give you an idea of where we've worked over the last 20 years, this map, you can see a lot in the mid-continent, drifted off in the front range of the Rockies, up into Nebraska, Missouri, even up into Iowa. Currently working on two big sites down in Big Bend, Southwest Texas, an enormous rock shelter, and GLD, which is an open air site with a Clovis camp buried in it, the first of its kind in that part of Texas. And then you see, kind of sticking out all by itself, Bluefish Caves. So, so we're gonna, as I promised, I'm gonna get to that in a minute. So this was a paper that was presented in Santa Fe in 2013, and some of you may know Stuart Fidel, and I like Stuart, but he gave this talk, Pre-Clovis and Bigfoot, the searches converge. Do you think he's skeptical about Pre-Clovis? Yeah. I mean, every time a Pre-Clovis manuscript comes out, the first person that comes out with a counter article is Stuart. Okay, but that's a good thing because he has to keep guys like me honest and make sure we've done our work. And I have no issue with that. I'm glad Stuart's around keeping us honest. Okay, so I don't have any issue with him. But the word that comes to my mind when I think about the search for Pre-Clovis is elusive. Webster's definition of elusive: hard to find or capture, hard to understand or define. I would say it's maybe not as elusive as Bigfoot. One of the rare photos of him, I think in Canada, if I remember right, this was a Canadian picture. But what I see, when I look at Clovis sites, there's only one in the Central Great Plains. Central Plains being Kansas and Nebraska. One Clovis site in a stratified context. We find lots of Clovis points. They're laying out on gravel bars. They're in farm fields, just Clovis point here and there. But as a site, there was only one. It's actually it's an Odyssey site, one of our sites. So, basically what I'm saying here is pre-Clovis culture deposits, like all archaeological materials, have passed through a geomorphic filter, such as erosion, sedimentation, soil formation modifies an archaeological record, and other geomorphic processes. You think about this part of the world, solifluction, frost heaving, moves artifacts, right? So if you're dealing with a sparse population left a sparse material record, which is what I think is the case, that material either gets buried and preserved or gets removed or gets moved around, modified. And so you have to understand context is really important. So, some say it's like searching for needle in a haystack. Sometimes I do feel like this guy. Because, they say, go out and find it. So where do you begin? Where do you look? But what I'm going to tell you tonight is there is a systematic approach. That the most effective way to search for pre-Clovis culture materials is to systematically study sedimentary deposits and soils of the appropriate age, especially in settings that would have been conducive to human occupation and other activities. In other words, what I say here is taking a geologic approach to doing this. So it's not just like the needle in the haystack per se, but there is a systematic way to do this. But even that, it's not easy. So the methodology, I've just got it summarized here. So I look at alluvial, colluvial being slope deposits, aeolian, windblown deposits that date between about 25,000 and 13,500 years before present. And this involves surveying outcrops. So I spend a lot of time walking along stream banks where I've got good exposures, coring and trenching, intensive radiocarbon dating and OSL dating, strong focus on buried landscapes represented by paleosols. Paleosols are old soils, dating to about 25 to 13.5. Revisiting stratified sites with long histories of human occupation, especially those dating back to the terminal Pleistocene, and looking deeper at older deposits, sedimentary deposits. Because oftentimes the archaeologists would stop when they got to Clovis and they would never go deeper. So those are the sites I often go back to and look deeper. When the remains of mammoth or other Pleistocene fauna are found in the region, I make an effort to visit that locality. So when I get a call, I get in the car or in a plane and I go and I check it out. Okay, this is really important. This is what I want you to remember. So questions should be addressed when there is a claim of a pre-Clovis discovery. First of all, are the reported cultural deposits artifacts, or are they geofacts or naturally modified bones? Because stones can be modified by natural processes and they can look like artifacts. The same thing with bones. The younger artifacts drift downward into the cultural component that's claimed to be pre-Clovis. So you might have a bone bed that's not archaeological, not cultural, but an archaeological site above it. And some of those artifacts might drift through various processes, mainly soil forming processes, down into that bone. and thereby they're not coeval. They're not associated in time, but can fool you. Did old charcoal move upward through bioturbation into a cultural component that is claimed to be pre-Clovis? So it can also go the other way. You can have old charcoal move up into something that's not archaeological, and you date the charcoal, oh, that dates to that time. Well, context is problematic there. Is numerical dating accurate? That's really important. It's not always accurate. And did older carbons such as coal or detrital organic carbon contaminate younger carbon? That was a huge issue with Meadowcroft rock shelter. There was claims that coal, which is in the area, got attached to the charcoal that was dated. That was eventually disproven. Paul Goldberg, who's a micromorphologist, looked at the charcoal under a microscope and determined there was no coal attached to it. But that could happen and contaminate what you're dating. I refer to this as my checklist. So whenever I find something that we think is pre-Clovis, it's got to pass every one of these. So I got to have a check mark on every single one of those before I'm going to make a claim that it's pre-Clovis. Some of my colleagues have not done that, and then they've faced the consequences. They become targets because, oh, you didn't resolve one thing or another that's on this list. So this is really important. So I'm going to go through, show you 2 sites before I jump into Bluefish to explain what I just told you. So this is the Big Eddy site. It's on the Sac River in southwest Missouri. And so here's Kansas, Missouri. This is the Sac River right here. Here's a close-up air photo. The Sac River makes a big turn here, and there's a big eddy right there, which is why they call it the Big Eddy site. And it's on this bend, and it's mostly gone now because of erosion. It's pretty much removed most of the site. But back in this would have been 2004, 2005, Southwest Missouri State University was excavating there. If you look carefully, you can see the river in the background right there. And there's a berm that separates the river from this portion of the site. And what happens is they let huge surges of water out of Stockton Reservoir, which is upstream from this, to generate power. And so the river comes up about 15 feet in a matter of minutes, and then comes almost to the top of that berm. Of course, it infiltrates through the berm, right? And so this was a bit of a challenge, as you can see here. This is what it looked like every morning whenever they did that. They didn't do it all the time, but when they did it, this is it. And so we'd have to pump, they'd pump all the water out. And of course, it was incredibly muddy. It really was a challenge excavating the site. The only thing that was happy there were these guys, were the crawdads or crawfish, or they call them bugs in this part of the world. There are lots of them. There are a lot of them. And in fact, when you're walking around, you see these hawkish chimneys because they burrow. Okay, they burrow down and they bring up material. Okay, so you see a lot of these in the landscape as you walk around the site. And so the site is actually quite remarkable. It has a complete archaeological record that goes from the oldest Clovis. They've got Clovis at the bottom, and everything post-Clovis is stratified through the site at Big Eddy. So the youngest cultural component, I think, dates to about 500 years ago to Clovis. And it has a really rich late Paleoindian component, which I'm pointing the top of. You can see a buried soil. The top of it's right there where it gets dark. So it's a buried landscape. That's actually the top of that landscape dates to late Paleo, 9 to 10,000 radiocarbon years BP. And so from the top of that, they've got lots of Paleoindian material, particularly a lot of San Patrice points. They've got middle Paleoindian right about there, right in here, and they've got middle and early Paleoindian from right in this part of the profile, all in a stratified context, okay? And so Odyssey, I said, well, it's clear that people like this location. They've been here from Clovis to almost yesterday. There was something about this site, this location, that really attracted people. Remember, I said to you, one of the things that I look for or study are sites where there's a people, the good record of human habitation through time. Those are the places then to go deeper. So I said, let's bring in Odyssey, and what we'll do is we'll go down, we'll start at 310 to 320, which is where they had the late Paleo, and just keep going down until we basically start getting into groundwater. And so that's what we did. So these yellow squares in the middle here are ones where we went down to about almost 4 meters below the surface. Then it started getting really wet because of the water table. And so this is what we found. What looks like an anvil stone. Right next to it, some what look like hammer stones, and a really distinct church spall. This is all fine grain, we call overbank deposits, vertical accretion deposits. It's silty clay. So it's been deposited by flooding, where water comes up over a former stream bank. This used to be a floodplain, even though it's about 4 meters down. That was a floodplain surface at one time. And so it was a grading by overbank processes. Now that's an important thing to know, because I can guarantee you none of what I'm showing on here on the screen could be lifted up and deposited in these overbank deposits. If we're talking about gravel, we had a gravel bed, a lateral accretion deposit's definite, but not in vertical accretion. So these were left by people, okay? Now, we got radiocarbon dates from charcoal that was scattered around these objects. Just a piece here, a piece there, not in a feature, not in a cultural feature, but floating around. Right next to them, we have several dates of about 14,700 calibrated. So, we're what? So, I always think in radiocarbon years, but so we're about 1,000 years, a little over 1,000 older than Clovis, right? Now, here's your test question. We're gonna see who's been really listening. This has been challenged. Why? We got artifacts. No one's arguing about that. In fact, everybody's on board with these many, I think for the most part, the artifacts. But it's been challenged. Why? Why would you challenge it? We got a date of 14.7, actually several dates cluster around 14.7. So why would you challenge this? I think that's some pretty strong evidence. Okay, who said that?
Charcoal?
Yeah, charcoal. So what's the issue with the charcoal? Floated or moved in a flood? Well, if it did, it would probably be coeval with that, unless it was really old charcoal on the landscape, which is, I guess, a possibility. But there's a more distinct problem that I'm hoping you were listening to me. The crawdads. Who said that? Who said that? Oh, you. So how the crawdads mess this up? What's that? When they're tunneling, what if there's just naturally occurring charcoal, which often you find in alluvium and flood deposits? They move old charcoal up into culture deposits. You know what? I can't argue about that. So it is not a solid pre-Clovis site. Maybe it could be, but remember that checklist. That's one I can't check off, that the charcoal was in situ with the artifacts. You see the problem? Okay.
Do those crawdads, do they leave trace fossils like
They do, actually they do, but the problem here is there's such strong soil development, it would have overprinted that, would have erased it. Because, we're talking 14.7, the soil is really well developed, and I couldn't make that. I'd like to make that argument, but the soil development is so intense that it would have erased trace fossils. But that's a good idea. I mean, we think about those sort of things a lot. Is there, look for Crotavina. Are there Crotavina, which are burrows that get filled or not? But when you're talking about something where you've got just so much soil development, it typically erases something that old. So yeah, it'd have been nice if we could trace out where the trace fossils are and where they're not, but we can't do it here. Okay, one more site, then we're going to jump into Bluefish. Rolfe Mandel site. Smoky Hill River Valley, western Kansas. So I live over here, and we're going way out in western Kansas. And this is the Smoky Hill River Valleys off to the north. These are the bluffs, or the side slopes from the bluffs going down towards the Smoky. And this site is right up on the top of the bluff line. And 2011, I was actually with the Odyssey team. We were only about 15 miles from this, looking at a site where some mammoth remains had been found. Like I said, whenever I hear about mammoth, I get in the car and go, and I did that. And the Natural Resource Conservation Service was putting in some terraces, agricultural terraces. And since it was federal money, the state archaeologist was there monitoring this. And as they're scraping, they took about almost half a meter. They're going through what's called Peoria loess, which is windblown. And we know that Peoria loess stopped degrading around about 13,000 years ago. And so they're almost a half a meter below that surface. And this is what they start hitting. And so the state archaeologist knew that I was right next door looking at this other site. He called me and said, "Hey, Rolfe, you need to come over here. There's something really interesting," which was good because we'd struck out at the other site. We couldn't even find the mammoths they were talking about. We found some remains of squirrel. That was it. So anyway, so I brought my team over here, and we excavated around this to expose the remains of this mammoth. And we actually ended up spending 6 years here. It was a disarticulated adult, okay? And so about 40 meters off to the side at the same depth. Now, remember, they graded this. So at the same depth, they exposed this concentration of artifacts. There were about 1,000 pieces, mostly debitage, of Smoky Hill jasper. Smoky Hill jasper is not a local rock. It's the closest source is a couple hundred miles away. So it was exotic material, clearly cultural, and clearly a concentration. And I thought, this is great because, if you're butchering a mammoth, right, you're going to occasionally go off to the side and sharpen your tools or make new tools. And this is a napping pile. That's how I interpreted it. It's a napping pile. So it is, and it was within 1 centimeter depth of that mammoth. So, there appeared to be a good relationship, context. It's all about context between the mammoth and this, what I was calling a napping pile. Well, there were lots of limb bones. They were not in very good shape because, again, we're only just barely half a meter below the surface. There's a lot of weathering, soil formation affecting these bones. But I dated the bone, 16,235 calibrated years before present. Clearly a pre-Clovis age. So this made me really excited, because I had a feature, I had bones. Then we found the second mammoth, right, a juvenile, right next to the adult. Again, disarticulated like the adult. So this even got me more excited, seeing these mandibles. Let's go back to the concentration of artifacts. So the archaeologist who was working with me, remember, I'm not an archaeologist, I'm a geologist. There's my disclaimer, because I was calling this a napping feature. So Jack Hoffman looked at this and he said, well, you look at it really close. It's got pretty sharp boundaries around it. It looks like a pit that was filled in with material. And then he saw what he thinks are fulsome, when they make the, what do you call it? Channel flake. Thank you. He said it looks like a fulsome channel flake in this assemblage. Wasn't sure, but he said, but he's an expert on fulsome. So I'm pretty sure it's a fulsome channel flake. Okay, so what do you see? But it's same depth as the mammoth. So what's the problem? Let's forget the fulsome. Let's say Jack didn't know what he was talking about with the channel flake. What if we didn't know that? What's the problem? It's the same depth. Okay, who else? Who can I pick on? Any idea what the problem is?
Somebody dug it.
What's that?
Somebody dug it down.
Someone, you dug it down. Remember I told you this was graded off? What if that is a pit? It would have gone to the surface, right? So it is not in stratigraphic context. You see what I'm saying? It is not a good association. I cannot prove that there's an association there, because if it's a pit, it may have gone to the surface. They graded it, and so it really corresponds to that modern landscape surface. You know, Folsom people, presumably, if Jack's right, dug a pit and for whatever reason, and then dumped all this material into it. So again, back to that checklist, right? See the problem? You understand how difficult this is to prove a point, and how you have to check off the boxes? Okay.
If it's a pit, it could just be a garbage dump.
Well, for them, it probably, if Folsom people were using it as a pit, for whatever reason, they were dumping it into a hole. I can't tell you why they would have done that. To me, it seems labor intensive, but I don't know. I guess the problem is I can't prove that it's not. You know, I may be sort of right that it was a napping pile. Though. I mean, I see Jack's point. It does, you know, with napping piles, it kind of flare out, right? So you'd see depotage kind of flaring out. And I don't really see it other than when they were grading, they knocked a few pieces of material out. But you see, but the bottom line is I cannot, if I publish this in Science, Stuart Fidel will have an article out the next day telling me I was crazy, okay? So anyway, enough said. Okay, so I'm hoping you understand the problem that we're up against in trying to prove a site is as old as we're trying to say it is. Which finally, I'm getting to what you guys came here for, right? Bluefish Caves. It takes a team to do this, and so I've got photos of my team members: Dennis O'Rourke, Lauren Norman, Justin Holcomb, who's my postdoc, Caroline Kozlinski, she deals with ancient DNA, and you'll see why we're doing ancient DNA with sediments, sedimentary DNA. And Lauriane Bergeon, who's, if you might know that name, she's French, but got a degree at the University of Montreal, studying the assemblage of bones from Bluefish Caves. She did that for her dissertation. And in fact, when she did her dissertation, her intent was to prove that it's not an archaeological site per se, that it's not Pre-Clovis. But the more she studied it, the more she realized she was wrong in terms of what she was interpreting. But I'm going to come back to that. I'll circle back to that in a minute. We were on the Vuntut-Gwitchin lands, and we worked very closely with the Vuntut-Gwitchin First Nation. And they've been wonderful, really great to work with. They're very interested in what we're doing. They're wonderful people. They've been so kind to us. We could not do this without them. And but really, what gets me excited is how excited they are about what we're doing, because they're learning. We're getting them involved. They're going to be out there this summer with us, actually working, excavating. So I owe a lot of gratitude to Megan Williams, who's the heritage manager in Old Crow. She's been incredible in terms of helping us. And the Yukon government, the archaeology group, has been wonderful. Ty Hefner we could have never done this without Ty and all the help we've gotten from them, particularly in creating a place where we could work on the site, a camp. There's just so much logistics in doing this. I could go on and on about that. Some of the other archaeologists at his office and the Polar Continental Shelf Program, again, they provided the helicopter for us. Working through Ty allowed us, they were dropping us off, resupplying us. So my point is, a project like this requires lots of people from lots of different places, and it all has to come together, and hopefully it works. And it has, so far, has been a great collaboration. I just can't thank all these other people enough for the help we've gotten. So to talk about Bluefish Caves, I want to put it into another context of Beringia. So how do you connect the Eastern Hemisphere here with the Western Hemisphere? How do you get any movement across that, well, what you have to do is this: cool the global climate by 6 degrees centigrade, build an ice sheet of late Pleistocene that spans the late Pleistocene 28 to 13,000 years ago, drop the sea level 125 meters, and then expose a continental shelf 2,000 kilometers from south to north. And that's exactly what happened. All of those came into play that created, formed the land bridge, the Bering Land Bridge, right? Which allowed the movement of animals from Asia into the Americas. And most importantly, I almost forgot to mention this, all these animals, it was a very rich environment with mammoth, with horse, with bighorn sheep, a lot of animals. You know, you think you're so far north that it'd be a horribly cold, just like an Arctic desert. It was just quite the opposite. We know this from the faunal record, from the remains of animals that have been found there, that it was in fact a very rich steppe environment that allowed animals to move from west to east, and presumably would allow people to move from, basically from west to east across that land bridge, right? So if you look at a map that shows archaeological evidence of the Ice Age dispersal of humans to America, Paleolithic sites dating from 34,000 to 14,000 years ago, you see there are a lot of them in Asia, and then now these are ones that have been reported in the Western Hemisphere. And there's Bluefish Cave. So you got a lot of people here. You got to get them across, right? So then what happens is you get Beringia, which is the land bridge. You get the land bridge here, and you get this. This would be all landmass on the other sides of these dots. So that connection forms. And so here's Bluefish Caves, and here's the land bridge with step tundra or mixed forest steppe, and here's the ice. So they're actually both north and west of the ice in a pretty comfortable environment with lots of food, lots of animals. We know that. There's no question about that. So this is a map that Jacques Mars and Moreland, they produced. It shows the general configuration of the Beringia during the last glacial maximum. So about 20 to 21,000 years ago, this gray area right here was filled in by land, making that connection. And this is a really interesting map right here. So Grant Zazula studied the Bluefish River and Porcupine River, and I'm just going to sum this up real quick, is that he's proposed that there was a big basin we call the Old Crow Basin. It was filled with water during the time that Bluefish Caves was supposedly occupied by people. So it doesn't look anything like that today. There is no lake. That lake eventually drained. But it was basically water that spilled from the ice front was way down here off this map and was flowing, filling these basins with water. So it would have been a much different looking environment then than it looks today. A big lake. And that might, think about it. If you have a big lake there, that might be another thing to take into consideration when you start thinking about human occupation there. So I'm going to give you a quick kind of a flyby of the site, the way it looks now. So this was a picture I took from the helicopter coming in, and you can see cave 1, cave 2, cave 3 is around the corner. We're just going to kind of fly around a little bit. There's the view of the limestone ridge. There's cave 1 right there. And there's a close-up. Here's cave 1 that Jacques Mars excavated. Here's cave 2. And here's cave 3. The arrow right here is pointing, you can kind of see it through the trees. That's cave 3. Most of the work they did was at caves 1 and 2, and a little bit of the work was done at cave 3. So here's a picture of Jacques Mars excavating Bluefish Caves from 77 to 87. I've never seen him without a cigarette in his mouth. Honestly, every picture I've ever seen, he's got a cigarette in his mouth. But anyway, he reported, finding bones, as you see here, that dated to 28,000 years before present. In an as he claimed, is an archaeological deposit. So you can imagine, this was back in the 70s and 80s. There's controversy now. Think about what it was like then when he made this claim. And here's his mammoth bone core, the one I just showed you. Here it is. It shows here's these three what he called bone flakes that refit. And so he dated the bone. He said that's an artifact 28,000 years ago in calendar years. And there are stone artifacts. There's no one has challenged this. In those you can see microblade core, A and B, and then the rest are all bureaus in the lower part of that image. There's no question, these are not geofacts. These are bona fide artifacts. The stratigraphic context is important. So they defined units A being the bedrock floor of the, call them caves, I call them rock shelters, really. They're to me, a cave is a deep interior. These are actually what I call small rock shelters or alcoves, but I'll use the word cave. So this is the bedrock floor, and then this is only about a meter thick. Defined a lower loess layer, a middle loess layer, and then a disconformity right there, and then an upper loess layer, and then this what I call this humus limestone rubble mix. You can see a very distinct boundary right here. So from the dating, these are Jacques-Mars dates, 25,000 from the bottom of this loess to about 14,000 right about there, and then 14 to 10 to there, and all of this humus is post 10,000. That's Holocene. Most of the bones and artifacts were found in this zone, what they call the herbaceous steppe zone. And then they saw a transition. This is from pollen data to a shrub tundra, or the burgeonable, to a boreal forest. This will be boreal forest in the Holocene. The mammoth steppe, the 3 pluses means lots of bones. 1 plus, the bones have diminished. To the top, he called it impoverished because there were very few bones. Okay, so this I just kind of distilled here what Jacques-Mars and his colleagues identified at the site. And they published 3 papers, Canadian Journal of Archaeology, a chapter in a book, another chapter in a book. And when all this came out, there was a lot of pushback, particularly from the Canadian archaeologists. And actually, they were not very nice to him. I understand why. I mean, I could see how that could happen. Because they could not fathom that there were people in the Americas, 28, 24, 20, 15,000 years ago. That just was, they laughed at him. At conferences, they literally laughed at him. And kind of made him a little bit of a bitter man, from what I heard. Because of that, and I understand that too, how that can happen. So he was not treated very well. So I mentioned Lauriane Bergeon in her dissertation that she was going to try to prove that there was not good context. It was not really an archaeological site, but more of a paleontological site. And by the time she got done, she did a 180. Said, no, this is an archaeological site because of this. She said she knows a lot more about taphonomy and bone modification than me. But when I look at those, I've seen butcher marks. Those look like butcher marks. They're nice V-shaped marks. They do not look like gnaw marks of carnivores. And this is what convinced her that this is an archaeological site, because that then makes this bone an artifact. And she then dated that bone. And if you look at the date down here, 23,500 years ago. That's, that is an artifact dating to 23,500 years ago. She dated another bone dated to close to 19,000 years ago, and again saw what she describes as butcher marks. I should go back. I'm going to go back just for a second. Okay, here. Whoops. The other point she made was where the butcher marks are. This would have been along ligaments that hold the tongue. And if you're going after tongue as a food, that's exactly where you would cut. It's the precise location. Particularly looking at the orientation of the cut marks, that's where you'd go after, being able to cut the tongue out. So that was another important point she made. So this article came out in 2020. Unresolved questions about site formation, provenience, and impact of natural processes on bone at Bluefish Caves, Yukon Territory. This was a challenge directly to Lorianne's paper that was published in PLOS One, saying that, and you notice I've highlighted something in red here. It says, this paper critically analyzes the archaeological record for Bluefish Caves by focusing on evidence for significantly disturbed archaeological context. There's that word again, context, and alteration of bone by non-anthropogenic processes. The context issue, I could see that to some degree if you're trying to look at artifacts and the relationship to bones. I can see that as being possibly problematic. But if you're dating a bone that is an artifact, then that context is a non-issue. But the argument here is those cuts, they're claiming those were produced by such processes as solifluction, moving sediments and moving bones, and that there's roof fall in the sediments, and they're grinding up against that, or grinding up against the floor of the rock shelter. Although I have issues with that because it wouldn't produce nice, perfectly aligned, perfectly parallel marks like I just showed you. But that's all right. I mean, we need to have critical assessment. And that is why I wanted to return to Bluefish Caves, because I really wanted to see, okay, what is the context? What does the sediment look like? What are the processes shaping that archaeological record? And so in 2019, I launched a very brief expedition to Bluefish Caves. And there we are flying in. That's all the way to get there. So Dennis O'Rourke, Lauren Norman, who's an archaeologist, she's Canadian, worked quite a bit in the Arctic. Lorianne, of course, and myself. So we went out there, and this was the objective of that 2019 expedition. Determine if there are intact deposits in caves 1, 2, and 3. If intact deposits occur, conduct limited archaeological testing. 3, collect sedimentary ancient DNA, what we call SEDA DNA samples from the cave deposit. And 4, what I really wanted to do was relocate cave 4, because Jacques St-Mars never touched cave 4. So if there are intact deposits, that's the place you want to go to start fresh and do it using technology that was not around when Jacques St-Mars was excavating the site. Come at it from a different perspective and a more contemporaneous, modern attempt to establish what's the context, down to the sub-centimeter using laser transits, looking at said DNA. It's coming at it from a lot different approaches, that none of which, unfortunately, were available to Jacques. So I was really excited about the possibility of relocating K4 and working there, if it looked good. So this is what it looks like. I'm standing up on top of, actually above Cave 3, looking out towards the Bluefish River. This is where that basin would have been. Grant Zazula inferred this basin from, there are strand lines, like strand lines from a lake. There are lacustrine, which are lake deposits that are preserved, remnants of it in this basin. So, and it's dated to the LGM. So that would have been, if you go back in time, you'd be looking out into a pretty big lake during the last glacial maximum. Doesn't look like that today, obviously. Okay, so caves 1 and 2. I was hopeful that I would see some deposits still there that we could reinvestigate. But as you can see, they excavated it all out, and what they didn't excavate was washed out. And so there was zero. Oh, I shouldn't say zero. There was just, that's it right there. That's all that's left of cave 2 or for that, and there's none in cave 1. So that was a bit of a disappointment. So cave 3, that's a different story because they barely touched cave 3, and I think it was because while they found lots of bones, lots of placing fauna, they didn't find any artifacts in cave 3. The artifacts, there were well over 100 in cave 1. Most of them came out of cave 1. Smaller number came out of cave 2. But none came out of cave 3. And so they left sediment intact here and also on the other side. And so we were with the Yukon archaeologists. And they said, look, we really don't want, this is all that's left of these 3 caves. And as you all know, archaeology is a destructive process when you excavate, right? It is. It's a destructive process. It removes that. And so they said, we really don't want you to remove this because this is all we got. And that's fine. But they said, well, you can do some limited testing right along this edge. Maybe cut back 20 to 30 centimeters into it to see what's there because they were interested as well as to what's going on. And so that's what we did. We knew that the radiocarbon age from that cave, from cave 3, spanned the period from 33,550 years ago to 10,000, about 11,000 years ago. So we knew we were in the right time. Shows the various taxon, bison, horse, mammoth, all came out of that cave. And so what we did was, you can see this line right here. The only thing removed was to the right of this line that we put in. So we took this part out. I'm sitting there staring down at Lorianne, who's found some bone. There was no shortage of bone. I'll show that to you in a second. But the stratigraphy was pretty much what I showed you in that photograph from Jacques St. Mars' work, where you have this humus, really thick humus layer with lots of clasts in it, and this loess over bedrock. So bedrock's, again, it's pretty shallow. Given the type of profiles I work in the mid-continent, which are 15 to 20 meters tall, this is a pretty small profile and a pretty small amount of loess in terms of thickness. But I didn't see 3 units of loess like they had from caves 1 and 2. It's just one fairly homogeneous unit, but lots of bones. Typically, they occurred on that contact with bedrock. So there's horse and caribou concentration, lots of concentrations of bone. There's a mandible of a horse sticking out of the section. There was a lot of the bones were horse or equus bones. And here's Dennis O'Rourke. Dennis is an expert in ancient DNA. And Lauren Norman, you can see they've got their gloves and masks on not to contaminate the samples with modern human DNA. And so for those who are not familiar with this process or think about how DNA gets incorporated into this type of sediment, basically all this organic material, as well as animals, what passes through animals, ends up in the soil as organic matter. And so I'm not about to go on the process of extracting ancient DNA. It's way beyond my wheelhouse. I feel a little bit even uncomfortable talking about it because it's so complicated. The bottom line is, if the DNA is preserved, there's a way to sort it out, and sort it out by genus and sometimes by species. Okay, so you can get a pretty good, and cold environments are ideal because the cold preserves that organic matter, which in turn preserves the DNA. So we're thinking this is the ideal place to look at it. Both look at plant DNA, what plants were growing, and what animals ended up in this rock shelter. So the stratigraphy I've got drawn here. So this right here is that right there. You can see this little bulge of humus going down into the loess is that bulge. And all these little boxes you can see sort of scattered where it says E in front of it. Those were all samples that were collected for environmental DNA. I'm going to come back to it in a second. So they take that, they take it to the lab. We have an ancient DNA lab at the University of Kansas. It's extremely sterile for good reasons, to keep foreign DNA out of it. That's Caroline. She's doing this for her PhD dissertation. This is part of it, studying the environmental DNA of Bluefish Caves. So what I'm going to do is I'm going to show you, just real quick, what we learned from all these samples that were collected through the profile. Well, first of all, they could not get DNA out of that humus layer except from one sample. They managed to get this E11 sample did, but this one did not. But all of the other samples that were collected in the loess yielded environmental DNA. So that was the good news. Only lost one sample in this process that turned out to be sterile. So that was very encouraging. What was also interesting was the richest part of the profiles were the lower parts, where they got the most DNA, the strongest signals, were all these ones you see that are circled. Those had excellent DNA preservation and were extremely diverse in terms of the animal assemblage. Very diverse. All right, so this is what an ancient DNA person generates. The diagram looks like this. So the sample numbers you see along the bottom are all the samples that yielded ancient DNA. All the ones that are within those boxes came in the lower part of the profile. And so these are what they call read counts. So the bigger the circle, the larger the read, which means more DNA came out of it. So you stand back and look at this, some really jump out. So, particularly, the ovis, equus, horse, ursus, which is bear, canis, which is dog, with wolves would fall in that category. And then there are some, you see a little bit of a decent signal of mammoth, but those really jump out. And you see a lot of, there's a lot of void space as well. So those bottom samples were extremely rich. And so looking at it in two fashions, both vertically and horizontally, again, what this shows is ovis, which ovis is sheep, right? Am I right? Yeah. Ovis is sheep, bison, equus, horse, ursus, bear, mammoth. I would have put a box here too for canis. Looks pretty good to me, but I'm not an ancient DNA person. So anyway, but the bottom line is, especially when you look at where they came from, these lower ones, it tells us there's really good preservation in the rock shelter, particularly in the lower part, which at the other sites is where most of the archaeology was found, was in the lower component of the loess. Most of it, not all of it, but most of it. So these, these are all the preliminary results, that show promise, from the loess. It looks really promising in terms of the degree of preservation. And so we're going to do the same thing as I'm going to show you in a minute. We have sampled. Now I'm going to, before I get to that, I don't want to be a spoiler. So this shows the plant. I showed you the animal DNA. And so it's kind of an odd figure, but the lower bar here is the percent read. And so what I think is really cool about this diagram, we're going from the deeper part, so these are the deeper samples, to the more shallow samples. We actually can see the transition from what was probably more of a steppe or grassland to, we start seeing trees and shrubs. These would have been, woody species, birch and shrubby plants, to a transition to mostly trees, picea, which are cone-bearing trees, conifers. So this in itself, I think, is a really interesting story of the change in the biome through time, going from more of a grassland to more of a steppe to more of a close what you have today as a boreal forest out there. So not only is the animal DNA telling an interesting story, but also really excellent preservation of plant DNA, telling an equally interesting story about the change in the environment through time at that site. So, K4. There it is. We found it right there. Here's K1 and there's K2, not very far from those. And so, as I was wandering around looking for it, this is what I saw. And I had read Jacques Cinq-Mars' papers and looked at some of his photographs. And when I saw this, I went, that probably is K4. Now, it doesn't look very impressive, does it? I mean, if you walked up to that, would you think that's a cave? Or just think it's kind of a crack in the rock, right? Well, this is what Cave 2 looked like when they started. There it is. You know, an area about this high, exposed, filled with sediment. So look at that, and then look at that. So that got me really excited because it told me that there's a high probability that this is filled with sediment. And that's what we want. We want sediment. We want something preserved in there to actually go in and test. So that made my day. Made everybody's day to see that. So in that initial 2019 expedition, determined that no intact deposits are present in Caves 1 and 2, but are in Cave 3. There are abundant fallen remains in Cave 3, but no lithic artifacts were found by us. There's excellent preservation of settled DNA and sediments from Cave 3, and Cave 4 was relocated and appears to have sediments stored in it. So the mission was accomplished. We determined Caves 1 and 2, can't do anything. Cave 3, they really don't want us to do anything. But Cave 4, they said, yes, you can excavate Cave 4. So the plan for 2020 was to conduct test excavations at Cave 4, but for obvious reasons, we were not able to go in 2020. So we said, let's do it in 21. We could not go in 21. But in 22, the doors opened, and we were allowed then to come into Canada and actually do something here at Cave 4. So this is a view of our camp that we set up with the bear fence around it for good reasons. And Ty Hefner is noted here. They really did a remarkable job of setting up this camp for us. It was a lot of work. And there's Ty. There's the crew. And here they are excavating right out front of. You can see the little opening, excavating out in front of it, screening the material that came out of it. There it is, getting a little bit bigger, using a total station that was sub-centimeter resolution. We also had a film crew. Yap Films was preparing a film for the Discovery Channel, which will be shown probably within the next month in the U.S., and also for the equivalent of PBS in France. They've already shown it. I've not seen it yet, but they were there with us for about 5 days during the excavation, the latter part of it, to do this documentary. So I'm anxious to see it. Of course, after my experience with the New York Times, maybe I'm not. We'll see. But anyway, also we had the Limtuk Gwich'in come out because we were constantly going back and forth with helicopter, and so we'd bring them out to the site. They'd spend like an afternoon with us, and they were really interested in what we were finding. And they're, like I said, some of them are actually working with us excavating in 23. So the stratigraphy, the important thing, this is what I wanted to determine, was the stratigraphy of Cave 4, did it look like Caves 1, 2, and 3? As you can see in this photograph, if you've been watching, there's that humus layer over the loess. There's a lot more colluvium, which is slope material, because it's real easy for slope material to come into this. And probably a fair amount of that is roof fall. But this is equivalent to Jacques Cinq-Mars' Unit B and his Unit C. If you can see all these little toothpicks, these are soil samples for both my work and for ancient DNA. And if you look, now the archaeologists hate it when I show up So I always mess up their profiles. I took out these big fist-sized chunks to do micromorphological analysis. So they always cringe when I show up. But anyway, so with micromorphology, these are not the Bluefish Caves. When I was out there doing it, it was raining and sleeting, so we didn't take a picture. The weather was terrible. But basically what I do is I cut out blocks like this. I take them back to a lab. We impregnate them with resin and cut thin sections that light can go through. You put them under a microscope. And you can see things that are really important at that scale that you cannot see with the naked eye. Okay, and this is important. So Justin Holcomb, who is my postdoc, he knows a lot about micromorphology, more than me. I know enough to be dangerous, but because it's a very specialized area of study. But here's what I just described to you. Here's a thin section. It's a glass plate that is sediment that was impregnated with resin and then you cut. And you can see all sorts of things in it that you can't see looking at a profile of the wall. Now, this could put you to sleep, but I'm going to try not to do that. So you can see such things, you can see such things as frost creep, shear zone. This is from solifluction, where it moves down the slope. You can also see soil structure. In this case, you can see this, it's sort of what we call platy structure, and soil carbonates that form in place. You need a fairly stable soil environment to get carbonates forming like that. And the takeaway here is that, let me show you here, is the upper 35 centimeters, there's potentially moderate frost creep up in this part of the profile. But once you get deeper in the lower half, it's relatively stable with minimal frost creep. There's frost-induced platy structure, which you see here, authigenic carbonate, you see here, which again requires stability. So why is this important to make this sort of definition of the soil, the profile? Well, what it's telling us is there's not a lot of movement in the lower part. And by the way, now I'll tell you, we found lots of bones. We did not find any artifacts yet, but we did not get to the bottom either. But at least tells us from 40 to 75 centimeters, and we're going to go down deeper when we go back, down to the floor. At least the bottom part is pretty stable, did not move much. Therefore, if we find artifacts, it's highly likely they are in association with any, and we've already got lots of bone, in association with bone. In other words, it has not moved. Conversely, anything in the upper part could be problematic because there has been frost creep. There has been movement of those sediments. So I could not have told you this just by looking at the profile with my naked eyes. With a micromorphological approach, again, Jacques Cinq-Mars did not have this tool. It produces a totally different story about stability versus instability. Do you see what I'm saying? Does that make sense? So this is going to be really important if we find artifacts. Lauriane Boujon, here she is looking at the faunal assemblage from our work last summer. There were a lot of bones, a lot of bone fragments, several hundred. And so she's been identifying them. And it reminds me a lot of what Jacques Cinq-Mars found. Not much in the upper part, pretty impoverished. Rodents, snails for the most part. Looks like maybe a caribou there. But in the lower part, quite a few animals, ranging from gastropods to birds to rodents, small-sized mammals such as hare and fox, medium-sized mammals such as caribou, and large-sized mammals, bison and moose. But you can see it gets progressively richer. Again, the circle indicates a greater abundance based on the size of the circle. So it looks really promising. All we need now is to find some artifacts to hopefully see if they're in place with the faunal remains. This actually shows the, by number, total of, oops, back up there. Here we go. So it shows the actual number. So the level 3 is the lower part. And so you can see it's, look at the numbers, you can see it becomes richer in that lower part of the loess. More diversity too in terms of the size of the animals. This is a really interesting diagram. So this is getting more into the taphonomy. So Lorian, what she did was, she's looking at all these remains under a microscope and looking for modification of the bones. And so if you're arguing that animals, carnivores, were dragging these animals into these rock shelters to eat them, right? You're going to see a lot of bones that have passed through the digestive system of those animals. And that leaves a telltale signature of digestion. And what she sees is this is none versus probable versus definitive, definite. This is a very telling diagram. You can see this is in percent. 80% of the bones showed no evidence of digestion. Probable, a little bit less than 20. Definitive, only a couple percent. So very few of these bones have been digested. Weathering, none is pretty high, almost 60%. Low, close to 40. High, like 1%. What this tells us is that the bone was not exposed very long. It was buried very quickly. Otherwise, it would show evidence of weathering, right? So that's important, which means carnivores probably couldn't get in there to chew on them, and scavengers couldn't get in there and mess them up. Abrasion, this is really important. Almost 80% of them showed no sign of abrasion. So that argument about moving and rocks grinding up against them, no. There's a low number, less than 20% abrasion, and high, like 1%. Coloration, this is just when you put bones in soil or sediment, they get discolored. So you see a lot of them. There is some coloration that's been imparted by the soil, but it's a pretty intense weathering environment in the boreal forest. So I suspect modern processes have affected that, the color of the bones. And again, root etching is pretty low. It's about 50%. So again, it underscores that they were not exposed to the surface or near surface for very long. They were the story that's kind of the takeaway from all this is there's not strong evidence of carnivore activity, and there's not strong evidence that the bones sat there very long before they got buried. So there's a lot of ongoing analysis. I'm going to wrap it up here. It's getting late. So we're radiocarbon dating of the fallen remains from caves 3 and 4. That's underway. Pollen analysis of sediments from cave 4, the ones I collected this past summer. We're going to look at pollen in those. And lithic analysis of artifact assemblage from caves 1 and 2. So we're hopefully going to go to the Canadian Museum of History where they're curated, and Cale Bruner, who's an authority on lithics from that part of the world, is going to hopefully be able to spend some time there and actually analyze the lithics using more modern techniques from Jacques St. Maur's collection. So what have we learned from both 2019 and 2022? Excellent preservation of DNA in sediments, page 3, which could help us reconstruct past environments. So it's a really powerful tool for paleoenvironmental reconstruction. Cave 4, we've tested it. We see that sediments have been deposited at depth in front of the cave. We never got to the bottom. So our strategy is to go back and hopefully go back into the rock shelter, get back into it this coming summer. The stratigraphy is similar to Cave 3 and probably similar to Caves 1 and 2. Preliminary fallen results similar to Caves 1 and 2. Lower part of the loess does not appear to be disturbed, and that's a really important takeaway. The future work, we're going to excavate inside the cave, more samples for soil sediment, DNA, pollen, micromorphological, and microdebitage analysis. Lots of lab work, radiocarbon dating, more pollen analysis, more fall analysis. So we're really this is going to take years for us to get through it, but very systematically. to see if we can tell a good story here. And I have no preconceived notions. Either it'll be, we'll see that, if we find artifacts, we'll see if they're in context. Maybe they will be, and maybe they won't. Some of the other objectives, what is the age of the sediments associated with fallen remains in Cave 4? So we want to establish a high-resolution radiocarbon chronology in order to assess the stratigraphic integrity of any cultural deposits that could be found in it. So, for example, I just sent 20 bone samples from our work this past year to University of California, Irvine, where they're going to do really high-resolution purified collagen dating on the bone samples. So we'll at least have about 20 radiocarbon dates before we even go back this summer. How did the plant and animal communities change over the past 40,000 years? Analyze plant and animal communities before, during, and after the last glacial maximum. How does the presence of a glacial lake affect the local environment? Again, look at plant and animal communities from a paleoenvironmental perspective to see if there are changes in the local environment that might be related to this glacial lake Old Crow. And and my colleagues are really interested in what were the caribou doing. So analyze caribou remains to investigate the origins of the porcupine caribou herd and compare seasonal range through the LGM. They obviously will do that through DNA analysis of the remains. So these are sort of tangential to the other part of the study I mentioned. And we're also going to submit a NSF proposal to look at the last 40,000 years using molecular methods. Bluefish Caves, Britannia Creek, sites in Hun Cave. We don't know the age of it yet. Little John site. So we're hoping NSF will support this through their Arctic research program. So we'll be able to tell a more robust story about this region over the last 40,000 years, but coming at it from a molecular method approach. And that's it. I want to acknowledge the Runtuck First Nation, Megan Williams again, Polar Continental Shelf Program, the government, the Yukon Government Archaeology Program, Ty, all of his crew members who helped us with all of this. Could not have been done without them. And with that, you guys have been really good. Hang in there. I'll take questions.